Hi everyone, this is the problem solving session 8 for basics of biology, and continuation with the last sessions. In this session also we are going to discuss about important systems of our body. So today we are going to discuss about circulatory system and muscular system, and so let's start. So the first topic that we are going to discuss is circulatory system. So, circulatory system refers to the heart and a network of blood vessels that move blood, nutrients, and oxygen throughout the body. So, why is it important? Because in order to be utilized, the food and water and air to be utilized in our body cells, they need to be transported to these different organs and different cell tissues. So the system which is responsible for this transport is the circulatory system. And as I mentioned in the first point, that this consists of the heart and a network of blood vessels, which can be further divided into veins, arteries, and the capillaries, depending on what type of blood it is carrying and the function of the site of exchange of these blood vessels for the blood and the organ. So in general, arteries, they carry the blood away from the heart. The veins, they carry blood back to the heart, and the capillaries are the site which facilitate the exchange of oxygen and nutrients to the cells. And this system as a whole helps tissue get sufficient oxygen and nutrients, and helps in also removing the metabolic waste products in complementary to partaking in circulating the blood. So just to show you on the diagram, so this is your heart and this is where the lungs are. So on this side you see the arteries which are carrying the blood from the lungs to the body, and this is a special kind of artery or special kind of vein, I should say. And and the main purpose of arteries are to carry the blood from the heart to the body, while for the veins it is from the what do you say, like from the heart to the lungs. So that's the main route of carrying of veins. They also bring blood from the body to the heart. So one is bringing the blood to the heart and one is taking the blood away from the heart. So that's the two main difference between the arteries and the veins. So moving on, there are different kinds of circulatory systems and different organisms. So the first kind is open circulatory system. So in this kind of circulatory system, you have blood which flows through lacuna, basically large open spaces, and through sinuses among the tissues. So there's no really like there is no proper vessels through which the blood will be transported. And because there are no vessels, there are no covering where the blood is being flowing through, these blood in this organism the blood flows at a very low pressure, and the tissues in these systems are in direct contact with the blood. This is in contrast to what we will be studying for the closed circulatory system, where you have a medium of exchange for nutrients between the blood and the organs. But here in open circulatory system, the tissues are in direct contact with the blood, which facilitate the direct exchange of nutrient and gases. And the flow of blood here cannot be stopped as it flows in the open space. So there is no restriction to the flow of blood. The respiratory pigment, which is because blood has to be carried everywhere, and blood has to be you will read, we'll see about it, that how there is something called hemoglobin in the blood, which is responsible for like carrying the oxygen. So the respiratory pigment in the blood here flows through the plasma. So it's dissolved in the plasma, which is a component of the blood, and it flows through that. And it is important because here what we are trying to highlight is, in open circulatory system you don't have red blood cells, but you have the respiratory pigment in the plasma. And such kind of systems are found in higher invertebrates like insects and prawns. Or so you find them in these kind of organisms. So you can see the diagram as well, where you see that there is a heart, and then you have these hemolymph or in the sinuses, which directly facilitate the exchange of nutrients and gases between the blood and the organs. And this is in contrast to closed circulatory system, where you rather have blood flowing through a closed system of the heart, of the chambers of the heart and the blood vessels. And as a result, the blood does not come in direct contact with the tissue. because they are flowing in what you call like a membranous structure, membranous tube. There is this blood are in high pressure, and here you have red blood cells in the blood which carry the component or the pigment, which is actually the respiratory pigment, which is responsible for carrying of oxygen to the lungs. And they are more efficient as the volume of blood here can be regulated by contraction and relaxation of the smooth muscles. This is what we were trying to compare with when we, in the previous slide, I mentioned about we cannot stop the blood flow. And the blood flow, because of being contraction and relaxation, it is quite rapid in the system. And as I mentioned, that there is a medium of exchange of nutrients and gases between the blood and the organs, and which happens via the capillaries. And such kind of closed circulatory system you will find in mollusks, echinoderms, and in all vertebrates. So basically, your heart is also a closed circulatory system. So just to show again, if you compare this to the previous slide, you can see that the heart gives out blood, but these bloods have the small vessels into which the blood flows. And because of being like how you say, because of being flowing through this kind of channel or membranous vessels, they are in high pressure. Okay. The other part of the blood circulation system is something called lymphatic system. So lymphatic system is mainly consists of the lymph fluid along with lymph capillaries and lymph ducts, which is an extension of the circulatory system. So if you see here, these are the veins and these are the arteries, but you have these lymphatic nodes and lymphatic system branching out from these blood vessels. So what do they generally consist of, or what do they do? So these lymphatic ducts or vessels, they are of course present in various parts of the body, and they also consist of layered vessels. They have three layers mainly, and they are mainly like veins. So the main fluid that flows through these lymphatic vessels are lymph. And what does this lymph consist of? This is so if you consider your blood minus the RBC, so if you remove the red blood cells from the blood, then you what you are left with is called the lymph. So it is a yellowish kind of fluid, and in that again you have these lymph capillaries. So these so whenever we will read the word capillaries, it is branching out of the main blood vessel or lymph vessel into some these small thin layered vessels. And so similarly in lymph capillaries also you have small thin vessels which are lined by endothelium, and these one end unites to form the lymphatic ducts and one end is blind. So if you see here, all these small parts, these are called lymphatic capillaries, and they are also in direct contact with the tissue fluid. So now let's move on to heart, and one of the terms that you will always encounter when you study the structure of hearts are auricles, and subsequently you will also read about ventricles. So first try to understand what is auricle and ventricle before we actually move on to the components of the auricles and what is the functionality of the auricles and ventricles in the heart. So auricle is basically referring to a structure of the atrium of the heart. So atrium means the frontal portion, the anterior portion. So atrial of the heart, while ventricle refers to the lower chambers of the heart. So in mollusk or in human, the lower portion of the heart is called ventricle and the upper portion of the heart is called auricle or atrium. And so in the auricles, you have less musculature, while the ventricles have more musculature. The main purpose of auricle is to collect the blood from the body, while the ventricles are responsible for pumping the blood to the corresponding part of the body. So, corresponding part means it's either sending the blood to the lungs for purification or it's sending the blood to the body organs for facilitating the exchange of nutrients and gases. So if you look at diagram of or a figure of heart, you will see that these upper portions are called the auricles while these lower part are called ventricle. We will come to these main important veins and arteries. For now, just remember the main artery for the heart system is aorta, while for the veins, the main vein for the circulatory system is the vena cava. So now that we know about chambers of heart, there so if you look into the evolutionary history of heart as well, you will see that there has been a gradation from chambered heart to three chambered heart and to ultimately four chambered heart. All this has happened during evolution to facilitate better mixing, a better segregation of blood in the heart, so that they are delivered to their appropriate places properly, to avoid mixing of the blood, and to facilitate greater efficiency of the heart in pumping the blood to different organs. So we'll start with the most basic form, which is a two-chambered heart. And you can imagine what would a two-chambered heart mean. Because we just read that there are two chambers mainly. So it only means that there are these two components in this individual form. So there are one atria and one ventricle. So this is seen in fishes. So in fish, the single atrium receives the blood, as what atrium does in general also, and then the ventricle sends the blood. Now in fish, in place of lungs for purification, you have gills. So in fishes, the ventricles send the blood out to gills, so that these are, so basically, ventricles, they send deoxygenated blood to the heart, to the lungs or gills for purification, basically to get oxygenated. While atria gets these blood from the body, and then it gives it away to the ventricle. That's the only purpose it has. Once it is purified from the lungs, then the arteries, you can see here, takes the purified blood and brings it back to the systemic capillaries. Again, you can see if their blood, these, what would these blood mean, that when they are blue or deoxygenated, it means that they have lost the oxygen. So they are more rich in carbon dioxide, which is the waste product of respiratory system. So these are rich in carbon dioxide, so they are supposed to be taken to lungs so that they are respired out of the body. That's the general flow of what happens during the circulatory system or circulatory motion. So this is two-chambered heart. We then move to something called three-chambered heart, and you can imagine if it is a three-chambered heart, there has to be two components, of course, auricles and ventricles, but one of these component will be present as in two, as double. So here we have the atria of amphibian receiving the blood, and here we have the atrium divided into left and right. So the right part of the atrium receives the deoxygenated blood from the body, while the left atrium receives. So just now you see that in the fishes, you had only one point of entry of deoxygenated blood, and that was moving out to go to the gills. But here in three-chambered heart, you have further bifurcated so that the heart receives both the deoxygenated blood and also the oxygenated blood, so that it is responsible for transporting these blood to different organs for different purposes. So here, if you see, the right atrium receives the deoxygenated blood and the left atrium receives the oxygenated blood. But see from what is the source or the source of these blood that they are receiving from. So the deoxygenated blood is being received from the body. So the body cells are respiring, they are giving out carbon dioxide, and then these deoxygenated blood are being received at the right atrium. While the left atrium will receive oxygenated blood, which is to be transported to the body organs. So they receive this blood from lungs, and then they further pass it to the body organs. Now, very important thing to understand is, ideally, when you are looking into the figure, this should be the right and this should have been the left. Meaning that this, if this is the right, this should have been the one that should have had the blue color for deoxygenated blood. But remember, when we are showing any diagram, it is not from what we are drawing as a perspective. When you are drawing, you should imagine yourself as being part. So if you want to understand it better, maybe just align yourself with the screen or with the book and try to keep the screen in front of you. So when you keep the screen in front of you, this portion will come towards your right, and then this portion will come towards your left. So that's what we are trying to portray. A simple key to remember is, when you're looking at heart diagrams, it would be opposite of what your handedness is. So if this is your right hand, the figure right hand should be, right side should be here, and if this is your left, so the figure left should be here. Okay. So now, as we have just discussed, there are two parts of atrium receiving blood from two different regions, and this is an amphibia. But both these blood get mixed in the ventricle. So there is only one ventricle, and both the atriums are pumping their blood into the ventricle, and then the blood is getting, as a result, the blood is getting mixed here. So this one muscular ventricle pumps the mixed blood out to both pulmonary and systemic. So systemic and pulmonary means two different words, which is important to be introduced here. So systemic means anything which is to be related to the body. So what is to be transported to the body? Which blood is to be taken to the body organs, or which is supposed to be spread in the body? The oxygenated blood, because you are supposed to take these oxygens to the organs for requiring the respiration. So systemic circulatory loops are the loops that refer to the blood vessels which carry the blood to the different organs. So these would be the oxygenated blood. Now the pulmonary means something related to heart or something related to lung. So what would be taken to the lungs? The deoxygenated blood. So the deoxygenated blood which had entered to this heart and has come out from the ventricles will be taken to the lungs. So this is the pulmonary loop and this is the systemic loop. So that's what they are trying to refer here. Now, so this in amphibian heart, you see a complete mixture of the blood in the ventricles. But when you move to reptiles, they also have similar three-chambered heart, except for the fact that their ventricles have this kind of septum. So there is only partial mixing or income, but even though the septum is incomplete, there is partial mixing of the blood here. In these regions, you can still imagine that there would be mixing of blood, but it will be less as compared to the amphibian heart. And similarly, you have the mixed blood being transported here and being transported there. Now we move on to the four-chambered heart, which is what is present in all the vertebrates and in the mammals, and we'll see how is that different. So, can you imagine what would have happened for it to become a four-chambered heart from three-chambered heart? The Q was in the previous slide. What could have given rise to the second chamber of the ventricles as well? I'll give you 5 seconds to maybe imagine how evolution works. Did you remember that there was in the three-chambered heart we had two kind of hearts? One had complete mixing, one had partial mixing. The partial mixing had something called septum in between the ventricles, but it was incomplete. So you can imagine if the septum goes completely up and it reaches to this region here, then it would be able to divide the ventricles also in two parts. And that's exactly what happens here. So you have two ventricles and two atriums, left and right. So this is seen in birds and mammals, and this is important because it allows complete separation of the oxygenated and deoxygenated blood. So you can imagine there is absolutely no mixing. You have these bloods completely separated by compartmentalization, and they're taken to their respective location for being acted upon. And this is required because it supports a fast metabolism in homeotherm, because there is no mixing, and you in homeotherms you need to maintain your body's conditions according to the temperatures outside, and you need to maintain the homeostasis. So you can imagine this absolutely the absolute importance of this kind of system where you have no mixing of blood. So what happens in this kind of heart is, or the heart where regions here is, so first the atria, because the atria receives the blood, so the atria will contract. It will force the blood into the ventricles. The ventricles will then contract and force the blood through the arteries to the lungs and the rest of the body, right? And there will be so this will continue to happen, and the blood will keep going back. So the heart will contract, blood will flow out, heart will relax. Again, the blood will flow in, heart will contract to push the blood out of the heart, and then the heart will again relax. And so as I've already mentioned, that it permits really high effective supply of oxygenated blood to all body parts. So if you want to see the structure of the four-chambered heart, you can see that you have these right atrium. Again, remember the handedness. So this is the right atrium, this is the left atrium, right ventricle and left ventricle. Now from the left ventricle, you have the main artery, which is supposed to take oxygenated blood to all the body organs. That is called aorta. That comes out, that is coming out, which is further like it would further extends to arteries, and then it further gives rise to capillaries. Right? From the left ventricle, you have the blood moving into the pulmonary artery. I'll come to this artery and artery and vein in a moment. But the main purpose of deoxygenated blood is to send to lung, but it will receive, this side will receive from the body at atrium. So the atrium here receives the deoxygenated blood from the body, while here the right atrium receives the blood from the lung. So to again give it a very clearly, right side of the heart, which is this side, here the blood is received from the body and the blood is then passed on to the lungs, while for the left side of the body or heart, you have the atrium receiving blood from the lungs, and then the ventricle is passing the blood to the body. Now, so we saw that arteries mainly refer to oxygenated blood, meaning that it is being carried to this body, and veins is where which carries the deoxygenated blood. So it is to the heart from the body. But when you have these movement from the lung to the heart, you have the opposite. So pulmonary artery should have carried the oxygenated blood, but pulmonary artery carries the deoxygenated blood from the lung, from the heart to the lungs, while pulmonary vein, which ideally should have taken deoxygenated blood, it carries the oxygenated blood from the lungs to the heart. So this is important to remember about pulmonary artery and pulmonary vein, arteries and veins in general, left and right side of the heart in general. Okay, so this is a summary slide. So we have already discussed this multiple times, so I'll just go over in very quickly. So the fishes, they are two-chambered heart. So they are only this one ventricle and one ventricle and one atrium, where they receive the deoxygenated blood, which are then further purified at the gills and then transported to the body organs. Then you have the three-chambered heart in amphibia, which allows for complete mixing because of one ventricle, which receives blood from two atriums, left and right. But this ventricle has partial septum in the reptiles, which allows for only incomplete mixing of blood in these systems. While in the mammals, there is complete division between the left and the right. So you have two atricles, two atriums, and two ventricles, which which supply blood according to their needs. Okay. Now, moving further into it, so what are the components of circulatory system? So we have already seen that it is heart and blood, which is mainly constituting the circulatory system. So to define, heart is a muscular organ which is located roughly in the body's middle line, so basically in your thoracic region, and it is responsible for pumping the blood. While blood is a fluid connective tissue which plays an essential role in circulatory system as it carries oxygen and nutrients to the body, and it also regulates the temperature and acidic balance of the body. Now, to understand the flow of heart, exactly how blood flows through the heart, we'll see from here. So we'll start from here. So the blood enters the right atrium from the superior and the inferior vena cava. So the main vein which gets the deoxygenated blood from the body organs are the vena cava. So the heart first receives the blood from the vena cava into the right atrium. Second, the blood in the right atrium will then flow into the right ventricle via a valve. So a valve is very important because what happens is you are trying not only to avoid mixing of the deoxygenated and the oxygenated blood, but you also want to prevent the backflow of the blood from one segment to the other. So you have these valves which are sensitive to the cues that they receive. So when auricles receive blood and the volume gets higher, and because of which the atrium walls are like they swell up, they give a cue to the these AB valve, which is located between the atrium and the ventricles. They get the cue on this mechanical signal and they open up, and the blood is then released into the ventricle. So now, from the right ventricle, there is contraction of the blood which forces the blood into the pulmonary valve, basically into the pulmonary artery. So you have here again a valve which is called the pulmonary valve. So now the blood will flow through this pulmonary valve into the pulmonary trunk. Then it will move to the so it is distributed here by the right and left pulmonary artery to the lung. So you have the pulmonary artery carrying them to the lung, where it will unload the CO2 and in return load CO oxygen. Now this was and then afterward this blood is further so lung, the blood is purified at lungs. Now from there the lungs will be again delivered to the heart so that they can will be passed on to the body organs. So now this blood returns from lungs via the pulmonary vein into the left atrium, which moves on from the left atrium into the left ventricle via again the AV valve. Then from the left ventricle, it will forcing the aortic valve open. It will move into the main artery, which is the aorta. From the aorta, the blood will then move into the ascending aorta, which will be further then carried into the different organs in the body, where it will unload the oxygen and then in return load the carbon dioxide, and this will again be returned into the vena cava. So that's the complete procedure of how blood flows in the heart. So, so just now we will look into the structure. As I've already described the functionality, now it should not be difficult for you to understand the where would the valve be located and what will be their function. So basically, if you look at heart, it has four chambers, auricles and ventricles, two of each, and then you have the outer covering, which is called the cardium, and epicardium is the outermost layer. Then you have the myocardium, and the innermost layer is called the epicardium, endocardium. And then there are four important valves. So the first is aortic valve. Now I will read where the functionality is, and if so, I have given this figure as well as this figure. For this figure, you can go back to the previous slide and try to understand where could this be located. So I'll try to read it and then you can simultaneously try to get where would this valve be present, and then I will point it out to you where is the valve actually present. So the first valve is aortic valve. This prevents the backflow of blood when it is pumped into the left ventricle, from the left ventricle to the aorta. So it is mainly from the left ventricle to the aorta. So can you guess which side of this heart will, or where would the aortic valve be present? So here, when it is being pumped into the aorta. Next, you have the mitral valve. Mitral valve is which prevents the backflow of blood. So why? First thing is, why do they prevent the backflow? How will they prevent the backflow? So try to imagine them as something like this gated channel. So on receiving these, let's consider this to be the valve between the atria and the ventricle. So auricles and ventricles. Now auricles are on the upper side of my hand. So let's consider like this. So on this side of the heart, the blood is received. As a result, because of increased volume, the filling of the auricle, this valve receives a cue and it opens up. So once it opens up, because of the increase in, because there was pressure which was increasing on the upper chamber, this opens up, the blood flows out. As the blood flows out, the pressure on in this upper chamber has reduced, and as a result, the valve will close again. So because of this closing of the valve, the blood that has just moved into the ventricles will not come back onto the auricles, and that's how it will prevent the backflow. So after ejecting the blood from one chamber to the other chamber, the valve closes, and that's why it prevents the backflow. Okay. So the mitral valve prevents the backflow of blood pumped from the left atrium to the left ventricle. So it is from the atrium to the ventricles. So can you take a guess where it could be? It is here. So from atrial to the ventricle. So here it is, the valve is located here. The next is pulmonary. Pulmonary valve prevents the backflow of blood when it flows from the right ventricle to the pulmonary artery. Here, should be here, because this is where your pulmonary artery is. So your right ventricle pumps your blood into this. And last is tricuspid valve, which prevents the backflow of blood when it is pumped from the right atrium to the right ventricle. So this is the tricuspid valve. So you have tricuspid valve, and then you also have mitral valve, pulmonary valve, and aortic valve. Next, as we have already discussed this, the arteries carry the blood which is rich in oxygen, while the exception being pulmonary artery, which carries the deoxygenated blood to the lungs, while the vein carry the impure blood to the heart from like to the heart for oxygenation, exception being pulmonary vein, which actually carries the oxygenated blood to the heart. So moving on, we have these nodes which actually maintain the heartbeat. So this is called the sinoatrial node, and they actually time the beats of the heart. What is beats of the heart? Every time the heart pumps blood, and the rhythm of the heart is maintained by these nodes. And how do they do it? So this is, let's consider this to be heart, and you have this sinoatrial node here. So this is sinus node here, and this is the atrioventricular node, the AV node. So these SA node, they cause the atria to contract, and then they send the signal to AV node where they ask the ventricles to contract. So the opening of each valve is controlled by a signal. So you can see that this is a signal which is being passed from the upper sinus node to the atrioventricular node as and when the blood flow progresses. So you can see the placement as well. So this is the sinoatrial node, this is the atrioventricular node, and then you have these region when they are passed into, and these Purkinje fibers are what will carry these signals forward. So now moving on to the next part, which is blood vessels. Like I had mentioned, there are of three types: artery, veins, and capillaries. So we'll first talk about arteries, which are also further differentiated into arterioles. So you can see in this image very nicely it has been shown that this is the artery. They are finally further divided, branched into fine vessels, which are called arterioles. So the if you have to see the difference between them, so arteries are the muscular wall tubes that mainly carry the oxygenated blood, while the arterioles, these are the small branches of the arteries that lead to the capillary. The arterioles are very thin, very less in diameter as compared to the arteries, and that's why they have thinner walls while the arteries have thicker walls. And as we have already seen, the artery carries the blood from the heart to different organs of the body, while arterioles, they are just carrying the blood, whatever artery is being, like whatever is being flown through artery. So they regulate this blood pressure and blood flow. Similarly, veins are also further divided into venules. So here you have veins, and then you have these small branches which is called venule. So if you have to differentiate them, you can say by vein being a larger blood vessel, while the venule is a smaller, minute blood vessel that drain blood from the veins into the capillaries. And again, being of smaller size, they have smaller diameter than veins. And the structure is also different. So the external wall is called tuna externa, and the second layer is called tuna media. So both these layers are thinner in venules as compared to veins. Both these arterioles and venules lead to capillaries, and capillaries are the fine section vessels which are actually responsible for facilitating the exchange between the blood and the organs of the nutrients and gases. So they are very tiny. And how tiny they are? They allow only one red blood cell to fit through them. So you can imagine the diameter to be so small that there is only one red blood cell that can fit in it, and they allow the exchange of oxygen, nutrients, and waste material between the blood and tissues. And so the single layered endothelium, this is a layering of this capillaries surrounding the basement membrane, makes capillaries a bit leakier. So what happens in this is you have these kind of slits here, so they flow the blood. So they have this leaky behavior where you can have these things moving out from the capillaries, and because of this leaky behavior only, you have the exchange of the oxygen and the carbon dioxide at the site along with the nutrients. So now let's move on to blood. So blood is a fluid connective tissue. It mainly consists of three components or four components if you break further. So first is plasma, then we have platelets, and we have blood cells, which can be red blood cells and white blood cells. And these circulate throughout the body. They deliver the oxygen and nutrients, making up to 5 to 6 liter of the blood. Like an adult has 5 to 6 liter of blood, which makes up 8% of the body weight. So if you have to see the component of the blood, you can have, you can see here that the plasma. So if you separate this blood, or if you centrifuge and you separate these components, you see plasma being at the top. These plasma consist of amino acids, all the waste, electrolyte, gas, and different proteins. And then you have these red blood cells here, which will, and in between you have the platelets and the white blood cells. So the other elements will be the red blood cells, the platelets, and white blood cells, which will also be further five kinds, again based on what kind of cells they are. So, like I've already mentioned, the plasma is with what we consider without the red blood cells. So, it is yellow in color and it consists of all the salts and nutrients and water enzymes. And the main proteins which are present are the serum, globulin and serum albumin, and fibrinogen. And so, serum Serum is again different from plasma. So the serum is the contains only globulin and albumin. So if you want to differentiate between the serum and the plasma, so plasma will not be a plasma will consist of globulin, albumin, and fibrinogen, while the serum will only consist of globulin and albumin. Basically, serum is plasma minus clotting factor, and plasma is blood minus red blood cells. So red blood cells, moving on to red blood cells now. So red blood cells are biconcave cells. They don't have nucleus, which they lose as maturity, and they mainly function to carry oxygen. And how do they do that? They do that with the presence of a respiratory pigment, which is called the hemoglobin. So this is hemoglobin, and you can see that on the hemoglobin there are four sites where it can bind to the oxygen. So once it binds to oxygen, it is called oxyhemoglobin, and that's why your blood is so important, because it can carry four molecules of oxygen at a time. And it is there is iron which is present on the hemoglobin structure, which is important for the facilitating this binding to the oxygen. And this iron is recycled in the liver and spleen once the red blood cells gets perished, when the red blood cells is like they're signaled for death. And the production of rbc is also regulated by kidney due to the presence of hormone or synthesis of hormone, which is called erythropoietin. Then we move on to white blood cells, which is mainly important for cell defense. They consist of these different kind of cells, which are monocyte, eosinophils, basophils, lymphocytes, and neutrophils. And what they mainly do is because they are able to ooze out of the capillaries, they can reach the tissue which are damaged, and they also reach the lymphatic system, and they circulate in the body, ready to defend the body against foreign particles. And that is what we had read in the previous sessions as well about regarding immune system, where we had B cells and T cells circulating in our body uh, as part of cells body's defense. Again, it was divided into two, which is called what is called innate immunity, which is something which we are born with, and that's why it's a rapid response. It consists of these different kind of cells, where we see eosinophils and basophils, along with other cells like dendritic cells and macrophages. While we have adaptive immunity, which is acquired immunity, so these mainly are facilitated by the B cells and the T cells. B cells have the antibodies which specifically target the foreign particles and kill them off. And they are being helped by the T cells in the form of the helper T cells or the cytotoxic T cells. So this was the overview of immune system, where we had also looked into the antibody structure and how the antibodies consist of the light chains and the heavy chains, which bind to the antigens via the epitope and paratope interaction, and how we had also seen there was a lag period, which is when the antigen is being recognized and against which the antibody will then be synthesized and proliferated, so that there is one which leads to being the memory cell, which will retain the memory of the epitope, while the other half will be responsible for the active defense and killing of the foreign particles. Then we move on to the next part, which is platelets. And platelets are mainly responsible for the clotting of the blood. So any tissue, at any side of the body, whenever it leads to bleeding, you will see, you would have also seen there will be clot formation. So the clot formation are the hardened cells that you see at the site of your injury, right? That is called clot, and that is done by these platelets, which are also called thrombocytes. And how do they do it? So basically platelets are these disc shaped cells here, and they regulate blood flow when any part of the body is damaged. And so they are derived in the bone marrow via megakaryocytes, and they have very little span, like they have a lifespan of 10 days. And because of the presence of platelets, you have, and another important factor which helps in platelets, along with platelets, are the vitamin K. So these prevent bleeding or hemorrhages. And what do they generally lead to? Clotting, or basically they lead to coagulation of blood during a cut or wound. So you can see that here being a cut, you have the erythrocytes and the platelets moving towards the site of the injury, where the fibrinogen gets converted to fibrin and the platelets get activated to form a mesh-like structure at the site of injury, which is called the blood clot. We'll see that here. So you can see that a platelet gets activated to form a activated platelet, which forms the fibrin, and the fibrin is what forms this mesh-like structure at the site of injury. So in detail, what it means is that when an injury is happening, it stimulates the platelets to release various factors that initiate the blood clotting cascade. The first step is platelet activation and aggregation. So the blood clot is actually, as I mentioned, a network of these fibrin threads. And in this network, there would be deformed and dead dead cells of erythrocytes, leukocytes, and basically RBC, WBC, and platelet. The thrombin converts the fibrinogen. So the activated platelet leads to the thrombin activation, which converts the fibrinogen to fibrin, and this fibrin is what will form the mesh-like structure. So here, even further, if you want to study it better, it mean so what happens is when the collagen is exposed to platelets during injury. So collagen is here at the site of injury. So when the collagen is exposed to platelets, the platelets will bind to the collagen via surface receptors, which is mainly via the von Willebrand factor. And because of this, the platelets get bind to the site of injury, which is mediated by cross-linking and activation of platelet integrins. And because of this tight binding and aggregation, it will lead to the primary hemostasis. Then these activated platelets, which are at this site already being cross-linked, they will lead to the further activation of fibrin, which will form the clots here. That's how blood clotting takes place. Now, the other important related phenomena is blood pressure, and I'm sure everyone of you should know this at least by name. So I'm sure everyone knows that whenever you go for hospital, the first thing they do is they look at your blood pressure, and they have these like instrument which looks at your blood pressure, and then they give you numerics, which is somewhere like something like 120 by 80. So what does blood pressure actually mean? Just by name also you can imagine it means that the pressure or the force of the blood when it is moving against the arteries. And it is given as two numbers, 120 slash 80. Slash means these are two different counts for two different systems. The first part is called the systolic blood pressure, and the second part is called diastolic blood pressure, meaning the upper portion 120 is the systolic blood pressure, while the 80 is the diastolic blood pressure. So if you have to define systolic and diastolic, systolic means the pressure which is exerted when the heart is beating, when it is contracting. While diastolic blood pressure is the pressure which is exerted on the arteries when the heart muscles are relaxed, that is between the contractions. Normal range, as you know, that whenever you go to hospital, you are on also read in textbook, you find that the normal range for both of them taken together is 120 by 80. But if you have to further look into the details, it for systolic it is somewhere around 95 for the infants. For adults it is between 90 to 120, and for diastolic pressure for the adults it is between 60 to 80. So as I've already mentioned, ventricles contract during systolic blood pressure and relax during this diastolic blood pressure. And because it is contracting, the blood pressure is higher for systolic as compared to diastolic when it is relaxed. So based on this, you have some problems of heart. So you would have heard this term quite a lot of often, which is called high blood pressure. Can you now, based on what you just read from the previous slide, can you imagine what would a high blood pressure mean? Or how could that be possible? Blood pressure was the force of exerted onto the walls of the blood vessels when the blood is flowing. So when you say high blood pressure, what could that mean? So that would mean that the force which is being exerted on the wall is too high. So what this could be meaning is, when does the pressure gets higher? When there is like this. So imagine there is the blood vessels getting narrowed, and when the blood is trying to flow through these narrowed vessels, the pressure is getting higher, and that is exactly what happens in blood pressure or hypertension. So what happens here is the arterioles, they become narrow, which leads the blood to exert excessive pressure against the vessels walls. And as a result, because now you are receiving the blood at a slower pace, the heart starts to beat faster so that it receives more of this blood. And it also gets enlarged, which is called hypertrophy. And because of this excessive functioning, sometimes the heart can also rupture or fail. So a that is a side effect of high blood pressure, and this could be because of multiple factors. Some factors relate to the some are because of congenital factors, but some of them are lifestyle related disorders. For example, stress, reduce of physical activity, food like which are rich in sodium, or fatty foods, or tobacco and alcohol use. So that's why it is suggested to have these unhealthy foods at a very probably at not at all, but if you when you're having these foods, you should have it in a controlled manner. The next is atherosclerosis. Atherosclerosis is a special type of or specific type of arteriosclerosis. So we'll first read about arteriosclerosis and then move on to atherosclerosis. So arteriosclerosis is when the arteries become thick and stiffened. Again, when you can imagine that the artery is supposed to be allowing blood movement, but it becomes stiff. And because of it being stiff, the blood flow is getting restricted. So again, here the blood pressure, ultimately the blood pressure will lead to an increasing, the blood pressure will increase. But this hardening of the arteries happens over time. And when it is affected by a certain factor, that is like named according to the specific component. For example, atherosclerosis. It is because of the buildup of fats or cholesterols inside the artery walls. So here, if you see, this is the normal artery, but in the condition of atherosclerosis, you have these fatty acids and cholesterol getting deposited into the walls of artery. They over time they start forming plaques, which is called plaque formation. And as and when they start going up again, you can imagine the narrowing of the artery leading to exerted blood pressure. But as and when the plaque formation goes to a higher level, at some point it can also burst, and when it bursts, it leads to blood clots, which is called thrombosis. And thrombosis can have a very severe effect, which is called heart attack. So in this condition, because blood the heart doesn't receive blood properly because of clots, the heart fails to function, and this is what is called heart attack or myocardial infarction. And all this is because of fats, cholesterol, which is again a lifestyle related disorder. So you can imagine the importance of having a healthy diet in the lifestyle. Okay, so that was all about circulatory system. We now move on to muscular system, which is connected to locomotion and how the different parts of the body move. So locomotion is what we refer to as the movement, which is characteristic of all living organism, and which actually will refer. So movement and a locomotion differs by the fact that a locomotion leads to a change in place, and there are three type of movements. So there is amoeboid movement, there is ciliary movement, and then you have the muscular movement. So amoeboid movement, as you can imagine, this is from amoeba. Amoeba, as we know, that they have pseudopodia for engulfing their food materials. So these pseudopodia, like projections that move and engulf something, is called the amoeboid movement. And you have read this multiple time in about macrophages as well. Then you have ciliary movement, which is because of the presence of cilia. So you remember cilia are these small hair-like projections which move to a left or right and giving directionality to the cells. And we have read about this in the villi, in the digestive tract. We also find it in the epithelial lining of the trachea and the reproductive tract, as well as in sperms. Then we have the muscular movement, and as the name suggests, this is something to do with muscles. So these are not only about the muscles that are found in your legs and your hands, but it's also the muscles which are present in the interiors of your digestive system, hearts, and all the other locomotions. So we're going to study about muscular system as a whole. So locomotion, as we know that they are, so skeletal system, neural system, and muscular system together control locomotion, among which we are going to read about muscular system. So muscles are what constitute the muscular system, and the muscles, they originate from mesodermal germinal layer, and they have properties. So muscles are extensible, they are elastic, they get excited. We'll see how they get excited, and they contract. So they contract, they relax, they extend, they are elastic in nature. And you classify the muscles based on the location that they are present in. So they could be skeletal muscle, they could be visceral muscles, or also smooth muscles. And the third type is cardiac muscles. As you can imagine, this would be muscles which are present in the heart. So first is skeletal muscles. So these are striated. So striated means present like this. So striations. And voluntary muscles, which are associated with the skeletal components. So as you can see, these are associated with the skeletal system of our body. And these muscles are regulated by voluntarily by the nervous system. So they are mainly important for locomotion. So what that means is, so you can see they are long, tubular, and they have striated appearances, and they are voluntary. The most important factor is these muscles are voluntary. So you can decide when to move your hand and when to not move your hand. But this is in contrast to the other two muscles. For example, cardiac muscles. You cannot control the cardiac movement. That is happening at its own pace. So that's why they are involuntary muscles. They are also striated, but they are cross-linked. So they're in this kind of branching pattern. While the smooth muscles are called smooth because of the fact that they are not striated. So you can see that they have all these single cells which are fusiform in shape, and they are generally present here in the inner walls of the or of hollow visceral organs. So for example, in digestion, in small intestines, and in reproductions. And they are also involuntary. So to summarize, if you have skeletal, the body location is basically face or skin, wherever you have the skeletons. Cardiac in the walls of the heart, while smooth is in the walls of the hollow organs, which allow the passage of something. By cellular shape and appearance, skeletal muscles are single, very long, cylindrical, striated, and they have multinucleated, while cardiac, being striated and uninucleated and branching, so they have branching chains. While smooth muscles have, they're single fusiform cells, and they have no striations, and they are also uninucleated. About the regulation of the contraction, skeletals are voluntary, they can be controlled via nervous system, but cardiac muscles and smooth muscles are both involuntary. Cardiac muscle has its own pacemaker, which is the heart, so it will time the beats, while the smooth muscles are controlled by the hormones. But there is no voluntary part involved in this. So we'll do this in details now. So we'll start with smooth muscle first. So smooth muscles, as we have just read, that they are non-striated, and they are involuntary. So they, because we just saw that they will contract in response to any signals which are in the form of chemicals or electrical, which they receive from the nerves or hormones, for example, epinephrine and vasopressin. So these are controlled by hormones. They are structurally of two types: single unit or multiple unit. So single unit is basically like this one stretch of unit which has these single cells being connected by gap junctions. But multi-unit will have repetition of these kind of units across it. So you have these neurons which are getting like make at these junctions you have the neurons getting interconnected, and we've already seen that they have great elasticity because they're smooth muscles. They have elasticity, contractility, and the extensity, so they get extended as well. So that's the idea about smooth muscle. For cardiac muscles, the structural components shows that they are separated by something called intercalated disc, which you so intercalation means these are cross-linked, right? So the links wherever they are linking, there is what and the formations or the connection between those regions are called the intercalated disc, and they are darker in shape. Again, they are striated, so they have these multiple lines running through them, which are connected by gap junctions and desmosomes at these regions. And because they are uninucleated but they are connected to each other, they are that's why called syncytium. And they will be arterial syncytium or atrial syncytium or ventricular syncytium. And if you have to see them in a better like structures a little better, so you can see that these muscles, as we know, they are able to contract and relax. They contain, and they do so because of the presence of T tubules, and these T tubules are connected to sarcoplasmic reticulum. Sarcoplasmic reticulum has calcium ions. So, because of being connected to sarcoplasmic reticulum, which are responsible for release of calcium ion, these calcium ions are responsible for contraction, and as a result, T tubules are responsible or helps in the contraction of heart. And unit-wise, cardiac muscles are made up of cardiomyocytes or contractile myofibrils. Myo means heart, and as we have already seen, they do so the beating is at a regular rate so that the flow of blood is constant, and they are present in ventricles and atria, and they have their own mitochondria so that they can generate energy. So if you just see here, this is how the heart looks. One cross section of the heart will give you this kind of a structure where you have these different kind of muscles. And then if you pick one muscle, you see that they are multiple of these myofibrils connected to each other at location which are called intercalated discs, and you have their own mitochondria to generate energy. We then move to the skeletal muscle, which is what you use for your movement. So they are the most abundant muscles because they have all, wherever you will have bones, you will have muscles connected to it. So structurally, they are made up of muscle fibers, which are wrapped by these connective tissues here. So if you see, so these muscles, they consist of these muscles bundles or bundles of muscle fiber. These bundles are called fascicles, and the fascicles are what is held together by the connective tissue. Now, if you want to start looking into it, so first connection is called the tendon, and then you have these epimysium, the muscle fibers, which are organized into three layers. One of the layer is called epimysium, the inner layer is called endomysium, and the outer layer is called the boundary layer is called perimysium, which is this. So perimysium will be the boundary layer for each of these constituent. What are these each constituent? They are all fascicles. So what to understand it, if you see here, you have these multiple little blocks. These blocks are called fascicles. Each fascicles, if you look into them by zooming out, you will see that all of these fascicles again consist of these different muscle fibers. And one of the muscle fibers, if you look into with great details, you will see that they will consist of these small holes. We will see what are these. And these fibers, these fascicles are covered by or layered by something called perimysium. And this is bone. So the connection between the bone and the muscles are called tendon. So we were talking about muscle fibers. So this one of these dots are called muscle fibers, and we'll see how do they look. So these muscle fibers, again, if you want to move from the structure to understand them better, this is the tendon. Tendon moves to something called epimysium. These epimysium, then if you open them up, you will see that they consist of these different fascicles, which are layered by perimysium. If you open one of the fascium, you will see that they consist of the muscle fiber. So one of all these blocks are muscle fibers, and the muscle fibers are made up of these single lines, which are called the myofibrils. So to understand muscle fibers, what are these single component or single unit? These are long cylindrical cells which are also multinucleated, and they are lined by sarcolemma. So to see sarcolemma, you can come to the next images where you can see that this one layer, so this you can see this cross section here, and they are layered by sarcolemma, which is a plasma membrane. and the fluid inside is called sarcoplasm. Again, it consists of multiple nucleus, so it is syncytium, and it is also this also has its own sarcoplasmic reticulum, so it has its calcium storage organ, organelle. And these muscle fibers have these multiple myofibrils, which again run parallel in lengthwise. And these myofibrils, we will see that they consist of myofilaments. And the myofilaments, so each of these myofibrils, you will see here, they consist of again two type of myofilaments, which are thick and thin. So what are these thick and thin, which are important for the muscle contraction? We'll see in the coming slide. So the thin filament is referred to the actin filament. So if you see this here, actin is one of the major muscle protein. So you have the G actin, which is the monomer of this actin filament, and it is globular. So this G actin is the monomer. When it forms this long chain, it is called the F actin. On this F actin, so these two F actins will coil around each other, giving rise to this alpha helix, which will be around 14 G actin coming together. And once they are connected to each other, they will be then associated with troponin and these long tropomyosin. So this troponin and tropomyosin are found on the actin element, and these are important because they will be associated with the myofibrils for the time when they are not contracting, and we'll see how. Because troponin will also be masking the active site for myosin. We'll see how myosin is important for contraction. Because myosin is the next filament which is important, which is called the thick element. So you have these myosin filaments, which are divided into three part tails, the heads, and the shortened necks here. And these monomeric unit of the myosin are called meromyosin. And these heads, they have one, they bind to the thin filament, and they also have ATPase activity. So they can drive energy by hydrolyzing the ATPs. And so you can imagine that it has its own energy source, and it binds to the actin, which is a part of the muscle fiber. And so it says here that carboxy terminal of myosin filament meet together at H zone and N terminal meets at A margin. So we'll come to this uh, unit in the next few slides. Just keep this in mind that we have the actin and the myosin filament on the muscle fibers. Like they are present in the muscle fibers and they are responsible for contraction. But before going to do that, we will read about the accessory proteins or the other proteins which are associated with these filaments, which are tropomyosin and troponin. So the tropomyosins are these filaments which are like these thin filaments which are running parallel to the effectins, and troponins are these proteins which are present at certain sites, and they basically are those sites which are responsible for binding to the myosin. So basically they are masking the myosin binding site. So in normal condition, your actin and myosin are not really being interacting with each other, and they're regulated by these tropomyosin and troponin. Other than that, you have desmin, which is again present at Z line, and alpha actinin, which is present in the which is also present in Z line. So we'll see what does it mean. So now we come to this, which is called sarcomere. So if you see this as a myofibril, muscle fiber having myofibril, in the myofibrils you have these sarcomeres running almost contiguously for 100 repeating units. So sarcomeres are connected end to end. So one sarcomere is this one unit here. So this one unit is the unit of contraction. So whatever contraction happens, it happens at this one unit here. This unit are all contracted together. As a result, your muscles are able to contract. Now we'll do this structure in detail. So here you can see, so basically this sarcomere will have repeating units of actin and myosin. Where and how, we'll see in a while. So just to understand it very broadly, the sarcomere will have A band, I band, and A band, I band, and H zone, and there are these two lines, Z line and M lines. So if you see this structure here, these filaments here are actin filaments, and these red filaments are the thicker filaments are the myosin filaments. You can see that the end of the sarcomeres are marked by the Z line. These Z lines. So this is what we were talking about here, that these Z lines have the polymerization of desmin, and they also have alpha actinin. So you have these two proteins present here, which are responsible for the attaching of the actin filaments back to back. And then in midst of these actin filaments, you have these myosin filaments, and these myosin filaments are held together at a very thin membrane, which is called the M line. Now, moving on to the bands, so you can see that there is so the units are not completely overlapping. So there are some regions which only consist of the thin filament. Some region consists only of the thick filament, while some region consists both these overlapping regions. So you can, on the basis of that, we have these different bands. So the first is I band or isotropic band. So this is a light band because it only has actin filaments, as you can see here. So of the adjacent sarcomeres, the actins are present only in this band. So there is no overlap, so there is a light band. Then you have the A band or the anisotropic band, or this region to this region, as you can see. And you can see that there is an overlapping of the myosin and actin, or the thick and thin filament, because of which it gives a darker appearance. While the H zone, which you see here, in the previous slide we saw here, you can see that it is the only region, it is a region where you only see the thick filaments. So now coming back from here, you can see that when we go to previous slide, you have these dark bands and H bands. So now you can imagine what are these. These are all sarcomeres which are being connected to one another, and you have these dark bands and light bands. So it's these bands that you see here. So the sarcomeres are all present in this manner here, running across along this length. So to summarize all that what we have read till now, we'll start from the outer portion here. So you have these tendons. Tendons are connected to these muscles. Muscles have epimysium. When you see one cross section of the epimysium, you see that there are many fascicles which are lined by perimysium. You take one of the fascicles and you look at the cross section, you find that it consists of light bands and dark bands, and it consists of something called muscle fibers. And these muscle fibers have their own mitochondria, their own layering covering called sarcolemma, which surrounds the sarcoplasm. These muscle fibers again are composed of multiple of myofibrils. When you take one myofibrils cross section, you see that they have these sarcomeres, repeating unit of sarcomeres, and each sarcomere has its actin and or thin filament and myosin or the thick filament. So that's how you can imagine that there is so much of contraction that enables you to move your entire muscles together for whatever action you want to take. So before we actually move on to how the muscle contraction takes place, we're going to look into something called neuromuscular junction, and we'll see what is the importance of this. Yeah. So neuromuscular junction, as the name suggests, it is a connection between the neuronal cleft or the neuronal channel and the muscles. So what they generally do is, because we have seen, right, that the muscles are being controlled by nervous system. So for that to happen, the nervous system has to pass on its cue to the muscle, which starts or relays the information to the muscle to start the contraction, and that takes place at these junctions. So here, so to define neuromuscular junction, this is a site of chemical communication between the nerve fiber and the muscle cell. Now here, at nerve fiber end, you have these terminal branches where each terminal ends on a region of muscle fiber. So there are multiple regions where they end into, and because of which the signals gets amplified. Now on the end plate or the muscle, on the muscles part, it is called the end plate, where you have thousands of these receptors, and this will bind to the protein that is being released from the muscle nerve fiber. Now, after you have these are the terminal branches. After these messages or this impulse is being relayed to the muscle cells, this is generally in the form of a neurotransmitter, but mainly it is the acetylcholine. So it will bind to the acetylcholine receptors here, and then it will pass on the message. So here it's in the form of the flow of sodium ions, and because they are electrically so they are electrical in nature, there is movement of ions across these layers. So movement of sodium ions across these muscle membrane leads to depolarization, and because of the potential differences there is the depolarization, which leads to further changes in the muscle that leads to contraction. So just to see the nerve fiber in a better or in like in a whole, you see that this is called motor neurons. So they have their own cell body. The emergent nerve fibers are called dendrites. The main part of the long running part of the neuron is called axon, which is covered by squamous cells and myelin sheath, and then it ends in these terminals, which release which or which basically is in connection with the different points or different regions of the muscle fibers. So now we will see how muscle contraction actually takes place. So here, uh, in the relaxed state is what we have so far been studying. You have the Z line, H zone, I zone, and the A band. And so muscle contraction takes place by sliding of filaments, which was proposed by Andrew and Hughes Huxley. And the sliding is basically when there is an overlap increase in the overlap of thick and thin filaments. So these overlapping region right here, it the overlapping region gets increased. So you can see here, so it's being contracting. This region H zone is diminished because these filaments are moving closer to each other. So the region of overlap is getting increased, and when you can see when it is maximally contracted, the regions are almost overlapping in all the regions. So these, so you can imagine, right, that these were stretched like this and they're getting, so this distance is larger. When it is contracting, coming closer to each other, the distance getting shorter. And when it relaxes, it goes back to the original shape and the distance also increases. So this is what happens during contraction of muscles. So to see it in like exactly how it happens, the mechanism. So the first thing is the neurotransmitter is released from the nerve fiber, which initiates the muscle contraction. This is generally acetylcholine, and it is released in the synaptic cleft here. These acetylcholine receptor binds to its receptor, which is called the acetylcholine receptor, which is present on these muscle fibers, and this leads to depolarization of the sarcolemma or the plasma membrane. Now, acetylcholine binding leads to the opening of the sodium channels on these membranes, and the sodium channels leads to the influx of sodium inside the cell. Now, there is a influx of sodium into the cell, and it leads to an increase in the action potential. And this action potential will move all across the muscle fibers. And what it does when it moves all across the length of the muscle fiber, it leads to the opening of the sarcoplasmic reticulum. So the sarcoplasm has their own sarcoplasmic reticulum, which I had mentioned about them. So um, So as we so just to connecting from the previous slide, so the acetylcholine is released by the nerve fiber. They interact with the receptor on the membrane of the muscle fibers, which is the acetylcholine receptor. Once the acetylcholine receptor has bound to the acetylcholine, it leads to the opening of the sodium channel on the plasma membrane, and this opening of the sodium channel leads to influx of sodium into the cell. Now when this sodium is moving inside a cell, there is a depolarization, there is an increase in the action potential in the cell, and this move across all the along the length of the muscle fiber. On the way where it is moving along the length of the muscle fiber, it leads to the release of calcium 2 plus from the sarcoplasmic reticulum. Now calcium 2 plus does what is required for contractions. So when the calcium ions are released, the it binds to troponin. And what was troponin doing when we were reading about the structure? Troponin were binding to those sites which was responsible for interaction with the myosin, the interaction between actin and myosin. So calcium binds to troponin. Troponin undergoes an allosteric modification and it loses the affinity for thin filament. So now you can see, so this was how it was in the initial, right? That there was this long layer of actin which was bound by the troponin and tropomyosin. Calcium binds to troponin, troponin moves away, tropomyosin starts to slide, and then you have these myosin sites which are present on the actin getting exposed. As a result, the myosin now binds to the actin. So you see, there is this movement, there is this bending of myosin towards the actin. And on the way, how does it happen? How does this binding and this movement takes place? It also has an ATP binding domain, so it binds to ATP, hydrolyzes the ATP, uses the energy to bind to actin and slide. So it will slide towards the, so it will pull the actin towards itself. As a result, when it is pulling the actin towards itself, the muscles start getting contracted. So there are multiple myosins which are getting connected to actin and getting pulling these towards itself. So that is called power stroke. So you can see that the myosin filaments here will bend and pull the actin filaments towards the center of the sarcomere. And as a result, the bound ADP and the P, which was because of the hydrolysis of ATP, they are also released in the process. And as a result, your Z band gets shortened. So the Z band is the sarcomere's ending point. So the longer the Z band, the relaxed this sarcomere is. So because the Z band is getting shorter, your sarcomere is getting contracted. So myosin will not just remain attached. It will detach, bind to ATP, again hydrolyze ATP, again bind to actin, and it keeps pulling it inside because of the presence of calcium 2 plus in the cell. So you can imagine it is being constantly pulling the actin filaments closer to each other. So the frequency of contraction is quite high. But when does it stop? It just can't contract continuously. It has to go back to its normal state. So that happens when the acetylcholine cue is stopped. And when does that happen? When there is a neural signal which was initiating the contraction ceases. As a result, you have acetylcholinesterase which comes into play. Acetylcholinesterase, as the name says, it will lead to the cleaving of acetylcholine. Acetylcholine gone, there would be no binding between the acetylcholine and the receptor. As a result, the potential, the membrane potential which had been depolarized will come back to its resting potential stage. It will get repolarized. Sodium channels will close. When the sodium channel will close, there will be no more influx of sodium channel. Because of no sodium channel, the calcium will be taken back in by the sarcoplasmic reticulum. Because calcium has moved back, the myosin will also now release from the actin because the troponin and the tropomyosin will come back on the actin and bind at the same side where the myosin was binding. So as a result, they go back to the normal state, which is the relaxation state. So they go back to something like this. From this state where the myosin and the actin were in contact with each other, they go back to this form where they are no more in contact with each other. And this is where the muscle is said to be relaxed. Now, based on these frequencies of contraction and how does this contraction take place and how long do they remain, like how long are they in this contract position and when do they relax, there are different kind of contractions that happens for the cellular system. So the first is twitch contraction. This is a very short muscle contraction and this is very common. So you must have seen your eye twitching or somewhere like your some muscles suddenly start twitching. Twitching means something happening for a very small period of time. So uh, the mechanism is very similar, like what we just read about the calcium release and then the thick and thin filament coming into interaction with each other because of which there is contraction. But before it can contract to the maximal limit, it goes back to its it goes back to relaxation state. So here you can see, so this is how your normal maximum tension or maximum contraction takes place when it takes place in any muscle. So you have these period of contraction and then it begins to relax. But in pitching, you can see that the eye muscles and these cells, like for example gastrocnemius, these are cells which undergo twitching very frequently, and you can see the curve being very narrow because as soon as they go into contraction stage, they start going back to the relaxation stage. But in contrast to this kind of contraction, you have something called tetanic contraction. And now in this tetanic contraction, the contractions are so long and so frequent that you don't see the relaxation phase for each contraction. So what I mean is that the phase of contraction and relaxation for each contraction is not like it doesn't reach, it's not separated. So as and when it starts to relax, the next stage of contraction comes. So all these contraction take place with each other in very less time. So you can see in this curve, they are contracting very frequently, and only at very end it starts relaxing. So when it starts to relax, or when does this stable condition comes to an end, depends on the kind of stimuli it has received. But when it is prolonged, the contraction is prolonged, it is called tetanic contraction. Compared to this, we have something called concentric contraction, which is something when you, let's say you are pulling weight. So when you pull a weight, you are contracting your this part of the muscle. So this is called concentric concentration, where you have these muscles fibers coming closer to each other, and this is rapid because the length of the muscle is decreasing here. But so if we have to point it out once again, so concentric contraction release is generally, you can see it visibly in the biceps. So this is the upward phase. So if you pick a dumbbell and you bring it closer to your body, that's when you are doing contraction. And because when you're doing this, you are increasing the tension and you're shortening your muscle length. So there is a flex in your elbow. This is opposite to what happens when you are pushing this dumbbell back. So that is called eccentric, and that takes a longer time to happen. And because there you are your length of shoulder, these muscles are increasing. So this eccentric contraction again in the biceps is when you are the downward phase of an exercise. This is when the tension is so the it also produces tension, but the muscle is getting lengthened here, and that's why it is it's a slow process where when you are, so you will notice that you can do this frequently, but when you are doing this, it takes a longer time. Even if it is frequent, it takes little, there's a lag between these. So based on this, you can have these four kind of contraction. You can also have isotonic and isometric contraction. So isotonic contraction is what we just read about, concentric and eccentric. So this is when you can change your muscle length because of by exerting force. But isometric is when you are not changing the length of your muscle. So for example, you're just holding anything and you are keeping it in a steady state. So there is no change, there is tension still because you're holding something, so you're putting weight, pressure on your muscles, but you're not doing any kind of movement to increase or decrease the length of your muscle. So that is called isometric contraction, because contraction is still happening because of the weight that you have put on the hands, but there is no change in the length. There are also some disorders which happen because of the misfunctioning of the system. So there are some of these are very common. For example, muscle strain, which I am sure everyone would have heard at least once or have must have experienced as well. So when you overstretch your body or overdo any muscular activity, it can lead to strain. Just a normal strain leads to, for example, strain means tearing of muscle. So a normal strain which leads to pain and reddening and inflammation. But if that is at a severe level, sometimes you can rupture the muscle fiber, and that becomes really, really problematic. The next is tunnel syndrome. So this is something when it happens on your hands. So in between your carpals, you have this median fiber. So this syndrome happens when your median nerve becomes compressed because of the carpal bones. Why is that? Because when you are repeatedly using your hands for some kind of motion, you might end up giving more pressure on that median nerve fiber, and that median nerve undergoes compression, which leads to pain. So you can see, and then because it is a fiber victus, it is important for nervous corrections. This malfunctioning of the fiber or this compression can lead to something called numbness. You feel numbness on your palms or fingers. So that is carpal tunnel syndrome. The next is muscular dystrophy. This is a genetic disorder. What it means is, we have dystrophins here. So you can see the dystrophins are important for the interaction between the filaments and the um, neuromuscular junctions. But if you see here, these dystrophins are dysfunctional, and there are also many other proteins which are dysfunctional, because of which the muscles cannot act like properly function. And over time, that leads to muscle weakness and death of muscles and tissues, muscle cells and tissues. So these kind of patients are generally they are ridden on the wheelchair because they can't stand and they don't have the muscle strength. Then there is something called Myasthenia gravis, which is an autoimmune disorder. What is an autoimmune disorder? Is when you have some kind of antibodies or some kind of uh, like antibodies or some kind of fragment of proteins which target your own cellular component or your own body proteins. For example, here you have antibodies, so you have some antibodies which target the receptors of acetylcholine receptor. Now you can imagine if you have a receptor which has been targeted by the antibody, these receptors are not free to interact with the acetylcholine, and acetylcholine is what is giving the information for contraction. So there is no proper contraction of the muscle, so that this blockage of the receptors leads to again muscle weakness, and it starts by drooping of eyelids. The first muscle to get affected is these eyelids. So you can see they're starting to get drooping, and then overall leads to a muscle fatigue or muscle weakness. And the last is a neuronal disorder, which is the Parkinson's disease. This is because of a problem in the brain where you again these motions, muscular movements are gets affected because of a poor information relay between the nervous system and the muscular system. And signs of this disease are generally shaking, rigidity of the muscles, you have very slow movement, and you have postural instability. So you have problems while adjusting yourself. So these are the different disorders. There are many other disorders, but I picked the ones which are important to the course and important for you to understand as well. So that's all what we have read today. Now, on the basis of this, I've compiled some questions from the previous year's assignments. So I will read out the questions and give you maybe 10 seconds for each questions so that you can solve them, and then we'll discuss the answers. So the first question is, the heart pumps the blood fluid continuously through the body via which of these vessels? Next is the type of circulatory system which is seen in the earthworm. Is it open or closed? Next, the deoxygenated blood in fishes is turned into oxygenated blood. So basically, the purification of blood takes place in which of these places? And the next is the oxygenated blood and the deoxygenated blood in frogs, which gets mixed in which region? So this is basically the three-chambered heart, and you need to remember where does the mixing takes place. So these were the answers. So the first is quite easy. It says which of these vessels are mainly responsible for pumping the blood, which is blood vessel. Second is which type of circulatory system. So open was present in the invertebrates. In earthworm, you have the closed circulatory system. In fishes, in place of lungs, we had read about gills. So gills was the place where you have these purification of blood. And the fourth one, in frogs, you have two atrium but one ventricle. And the mixing, that's why it takes place in the ventricle when both left and right atrium are pumping their blood. The next set of questions are, so the first is, these have thin walls located closer to the surface of your body, and also walls that ensure blood flows in the right direction. So take the cue, it is in the right side of the heart. Next, the oxygen and nutrients are exchanged for carbon dioxide and waste at which site? So we have discussed this. Which among these is responsible for the actual exchange? Next, the division of atria and ventricles in a four-chambered heart is inward. So which part does what activity? Is it the atria receiving the blood and the ventricles pumping the blood? Or the ventricle receives the blood and the atria pumps the blood? Or it is the left and right division where the right atria receives the blood and the left atria pumps the blood, or the opposite? And the fourth is the normal heart rate for any AV node is beats. So beats basically how much contraction in 1 minute. So the answer to the first is vein, because it was right. So it was the deoxygenated blood part. So that was vein. Because the left side of the heart is concerned with the arteries and the oxygenated blood. Second is so it is a capillary where you have these exchange between the oxygen and the nutrients of between the cells and the blood, but not at the veins and the arterioles, but the finer capillaries. Next is the atria receiving the blood and the ventricle pumping the blood. The right and left division is for oxygenated and deoxygenated blood. And the normal rate for the heartbeat or the AV node is 40 to 60 beats per minute. The next questions are so the first is the oxygen carrying protein in blood is present in which of these cells? Next, the hormone essential for production of red blood cells is synthesized in which? So, this, if you could remember the hormone name, that would be great as well. And where is it synthesized? Next is the buildup of plaque in atherosclerosis occurs in which of these area or which of these places? And the next is the normal pressure in the arteries when the heart rests between the beats is one. So try to remember the blood pressure where we were reading about systolic and diastolic. When the systolic takes place and when does diastolic takes place, and what was the numerical value for it. Yeah. So the first one is oxygen carrying protein. Could you guess the name of the protein? Like which one was the protein that is actually carrying the blood? Hemoglobin. Hemoglobin had iron sites which were binding to four oxygen molecules, and that is present in the red blood cells or the erythrocytes. Next, the hormone which is essential for the production of red blood cells. And what was the hormone name? The hormone was erythropoietin, and they were being synthesized in kidney. Next, the buildup of plaque in atherosclerosis occurs in the inner lining of artery because it is the artery where it was being deposited, where the plaque is being formed, and then once it reaches to a greater buildup, can even lead to bursting. And the next one is the normal pressure when the heart is resting is diastolic pressure because it is between the beats. When the heart is contracting, it is the systolic pressure, and systolic normal is 120, diastolic normal is 80. That's why you the normal blood pressure people, the range is 120 to 120 by 80. Okay. Next is the cells that activate cytotoxic T cells to kill infected target cell in adaptive immunity. Even though we have not discussed this in this session in detail, but we had discussed this previously. So just try to remember what was the what could be the answer here. And next is the myelin sheath of the neuron that rotates around the axon for insulation is which of these cells? Next, the homodimeric protein of sarcomeric Z line that anchors the ends of affectin to its which of these? And then the striated appearance of skeletal muscle tissue is due to the repeating bands of which of these proteins? So the answer to the first question is helper T cells, as these activate the cytotoxic T cells in the immune system. The second is the myelin sheath, which is present on the axons are these concepts. We had seen this in when we were seeing at the figure. And the next one is the homodimeric protein, which was part of the sarcomeric Z line, is the alpha actinin. And actinin myosin repeats gives the striated appearances. The last set of questions are the type of contraction that occurs during the lowering phase of biceps. So again, try to remember when I was doing the action and we were reading those slides. The next is the neurotransmitter upon binding leads to the influx of sodium inside the cell during an action potential in the muscle fiber. So which one was the neurotransmitter here? Next, the muscle tissue which shows cross-linking stripes is which muscles show the cross-linking. And the next is the degenerative disorder of CNS or central nervous system that affects the muscular system. So, which one of these were actually the degenerative disorder for this central nervous system? So the first one is eccentric. When you are pulling it up, or when you are doing when you're putting it closer to your body, it is concentric. When you're putting it outside, when you're lowering it, it's the eccentric concentration contraction. The second, the neurotransmitter is acetylcholine. Third, cross-linking muscle tissue is the cardiac muscle. And fourth, Parkinson's disease, because muscular dystrophy, myasthenia gravis, and carpal tunnel syndrome is a biomechanic this problem. Myasthenia gravis and muscular dystrophy are different kinds. One of them has protein deficiency, the other is a problem which is related to the autoimmune disorders. Okay, so that ends up today's session. We have discussed muscular and the circulatory system. And again, if you have any doubts, you can post it in the comments section or in the discussion forum, and we will be able to help you. Thank you.